We started with trying to understand a human condition, a syndrome called sepsis, which is an organ failure system which is secondary to infection. And in order to understand what was happening, we developed a system that we call behavioromics. We utilize bioinformatics and we utilize digital analysis, and we hope that this will lead to us being able to use machine learning or even AI technologies, which will improve our ability to understand what the cellular behaviors are. This is how we differentiated how they begin, by watching what stages they start at, and so we designated those as different numbers, and in this case different colors. So it went from 4 to 8, 8 to 14, and then once it gets to 14, it doesn't stop. It just makes this huge cluster. And we discovered that part of that is molecularly based, meaning that it's a cascade of events which precede each other. So one event happens, which triggers the next event, which triggers the next event. And what we discovered is that you can impair that, and in this case we impaired it by a molecule called leukotriene, so that it goes from 8 and it tries occasionally to get to 14, but it kind of stutters. Instead of, it goes back and forth from 4 to 8, 4 to 8, 4 to 8, it kind of tries to get to 14, but then it degrades again. So this was really a big discovery, in that instead of moving to these huge pathological structures, we can actually intervene early and block it, so that this cascade is trying to advance, but we've impaired it. Hopefully this will also help us understand complex situations like sepsis and other multi-organ diseases, where we can pick and choose where we want to target and what we're trying to accomplish.